So welcome back, everybody. Thank you for joining us. So we now have an open session in which we will have an opportunity to hear from Brad Ringeisen first. Brad is the executive director of the IGI, and he will provide us with a general overview of what the IGI is about, how it was founded, its mission, its vision. And then I will briefly talk a little bit about Pioneer Science. It's specifically about the partnership with the IGI, but also touch upon a couple of other programs that we are developing. And at the very end, hopefully we'll have about 10 to 15 minutes maybe for general Q&A. So feel free to address Brad for questions about the IGI and myself as well if you're interested. Okay. So Brad, thank you so much.
All right, thank you. All right, excited to be here. It's really great. It's the first time in Brazil, first time in Rio. So I thank the organizers and all Sergio, Fernanda, Georgie, all of the scientists that we met today for giving their time. We've had a wonderful visit today so far, and I look forward to many additional interactions. And engagements between our it's not is it?
Yeah,
I can.
It's there.
Okay, there we go. So for those of you that aren't familiar with the Innovative Genomics Institute, or the IGI, this was an institute that was founded by Nobel laureate Jennifer Doudna back in 2014. Our building is on the campus of the University of California at Berkeley, but we work across a number of organizations. We work with University of California, San Francisco, which is a medical school, because Berkeley is not a medical school. We work with the University of California, Davis. It's an agriculture-focused school, one of the best agriculture schools in the world. And so we bridge across those two University of Californias in addition to Berkeley. And then we also work with the Department of Energy laboratories that are also in and around the Bay Area as well. The IGI is different than most institutes. Well, it's different because Jennifer founded it. She's pretty special. But it's also different because it's an institute that's founded with social purpose. We would like to develop technologies that could uplift humanity. We focus on large societal problems and to try to use biology and biotechnology to help solve those big problems. But we don't want those to solve problems that just affect the richest and those that are most privileged in the world. Jennifer founded this institute because she wanted science to help everyone, all of those people in the world. And so we say that we're founded with cutting edge science, but with a social purpose. We're looking at things in human health, which is why we're here today. But we also do a lot of research in agriculture and climate. But all of the research that we do is focused towards affordability, accessibility, and equity. And our partnership with Brazil and IDOR, I think, is an example of our desire to be able to make an impact across the globe. So what sets the IGI apart? Well, we foster collaboration. Jennifer and I sat down for a strategy session just a couple of weeks ago, and the word collaboration just kept coming up in what makes the IGI special. You've heard about it from our discussions today, as we have visiting fellows from IDOR collaborating with us inside the walls of the IGI. But we also do it across all of those organizations that we've talked about. And I think in academic research, all too often, professors and scientists just work on their own problem in their own labs, and they don't talk to scientists outside of their lab, outside of their department, outside of their institution. And the IGI truly is a collaborative organization, and we're starting to now bridge internationally across many disciplines and in many different topics. But we also bring a different philosophy. My last position, I've been at the IGI for about 3 years now, but my last position was with an organization called DARPA. This was a Defense Advanced Research Project Agency. It's a high-risk funding agency, high risk, high reward. What we did at DARPA was look at big problems and put large, big dollar bets down on her and who she is. And with my experience at DARPA, we wanted to look at trying to solve some of humanity's greatest problems. And that's really what we are going after now. We have longer time horizons. We're a philanthropically driven organization. So we don't have the requirements of short-term investments from those in startup companies or the reporting requirements that are necessary for a big government grant. The philanthropy allows us the privilege of working on these longer time horizon problems and applying science to try to make those impacts. We're also looking at scalable solutions, things that can actually go beyond just the laboratory. And I'll talk about many of those applications of how we can scale. We work with those individuals that manufacture cells for cell therapies. We work with agriculture, to try to actually scale some of those solutions out into the field. We're doing international field trials in Colombia now, and we're also looking at doing clinical trials around the world as well. And then finally, the last thing that sets the IGI apart is that we have a public impact team. This is a social impact team. These are scientists, but they're social scientists. They're scientists that are embedded with our research scientists to start thinking about the ethical implications, start thinking about the equity. What are the societal impacts to the groundbreaking science that we would do? And I think it's unique to have a team of scientists dedicated to thinking about that. They think about the regulatory issues, the ethics, as well as the communities around the world and around the globe that could be affected. And hopefully their lives could be improved by the technologies that we're developing. Now, what's wonderful, Sergio, Jorge, Fernanda, kind of looks like the IGI IDOR collaboration. Many of these topics are exactly at the point of intersection between IDOR and the IGI. Now, the IGI is pretty innovative. The first I in IGI is innovation for a reason. You all know UC Berkeley. It's a famous institute. I mean, it's a famous university. What you see here on the left-hand side is that the IGI alone accounts for anywhere between 12 to 15% of all of the intellectual property, all of the invention disclosures and patents across the entire University of Berkeley. It's an impressive number for just one institute and one center at the university. Now, I have to actually update this slide because we don't have 17 companies. We now have about 22 different companies that have been started just out of the IGI. They've raised something on the order of about $3.5 billion. Their valuation on the market right now is over $10 billion. And so part of the scaling that we do at the Institute is very much supporting entrepreneurs and starting startup companies that can have a big impact as you tap into private equity and private capital as well. But we want to do this with an eye towards equity. And for those of you that maybe have thought about starting companies, or maybe you've participated in starting companies, there's a big inequity when it comes to gender in entrepreneurship. There's very few women, led startup companies, and those that are leading those companies oftentimes are at a disadvantage for fundraising, and the numbers are really quite astonishing. So we have a donor, HS Chow, who has supported a new entrepreneurship program that supports gender equity. So we just graduated our first class of 3 fellows. I think 2 of those have moved on and won a $1 million prize that's non-dilutive. No equity was given to launch the start of those women started companies. Now we have a second class of 4 entrepreneurs that are starting just now in the IGI as well. And so this is really a big focal point, and who better to lead this than Jennifer Doudna herself. All right, so this is the IGI. We talk about genomic technology discovery and technology development. We're probably some of the best in the world in innovating across not just CRISPR, but innovating across technologies in plants, technologies in bacteria and microorganisms, different ecosystems. We're looking at natural lands that have been affected by forest fires and how to maybe try to have those lands bounce back more quickly. And we're just looking at a tremendous amount of genomic technology development across the primary living cells of human cells, bacterial microorganisms, as well as plant cells. But if it just stopped there, the IGI wouldn't exist. It would be individual labs. It would be Jennifer's labs. It'd be many of the different labs that are driven by different professors across our universities. But what the IGI does is drive those discoveries towards climate and sustainable agriculture solutions, and solutions driving improvements in human health and in the development of new diagnostics and new therapeutic treatments. And then we partner with different organizations, maybe one day partner with IDOR or the hospitals here in Brazil to do clinical trials and field trials. Right now, we work with UCSF and UCLA. We work with UC Davis. We also work with the CGIAR, which is an international agriculture organization that provides seeds to low middle income farmers around the world. And we are their innovation pipeline to be able to provide new technologies in agriculture as well. Now, we're here at IDOR. It's a health-focused and biomedical-focused institute. So let me just focus on our health strategy primarily today. We've discussed all day today a lot of gene editing innovations, but we also do target screening assays, and we try to discover new targets for disease. We also do delivery innovation, and for delivery, that is how to deliver genome editing tools more efficiently, more specifically to specific types of cells at high efficiency. Ultimately, moving on to systemic delivery or in vivo delivery of those reagents to be able to help drive down costs, which I'll talk about more later. We also have started a very large effort on microbial community editing, looking at organisms in the human gut. And we would like to try to look at editing and modifying and making changes in those organisms at the early stage of life, when the microbiome is a little bit more malleable, a little bit more changeable, so that you can potentially set a young child on a more positive health future. We also develop stem cell models and three-dimensional models and organoids and such. We just started a high throughput manufacturing facility where you can actually make different mutations of different types of cells, way beyond what you would be able to do just with graduate students or with human hands, because it's a fully robotic and automated system. And then we move on to validate if you can find a target, if you can develop a CRISPR modality or a genome editing modality of some sort. You've identified the cell and the tissue type. We then move on to do optimization of that, to be able to do in vitro and in vivo validation and proof of concept. We begin formulating, and then we start to scale up and move to translation. And the translation, again, it's partnerships with industry, it's partnerships with other organizations and other institutes, it's entrepreneurship and startup companies. It takes all of that. For different diseases, there are different pathways of success in these areas. Now, what types of applications, what type of indications, health indications are we working on? Well, initially, we're looking at things that are sickle cell and immune deficiency diseases. But in the midterm, we're looking at moving into more refined and more affordable ways of treating sickle cell disease. And then we're also looking at specific immune deficiency diseases. And I'll talk a little bit more about this, but there's one disease particular, Artemis skid, that is prevalent in Native American communities, specifically the Navajo tribe. And the Artemis skid is more prevalent in that community. So we're looking at a sort of a neglected and rare disease where startup companies probably are not going to go after a cure or a treatment for this disease, but we're a nonprofit. We're the University of California. We're philanthropically driven, so we're selecting this as something that we think is a very tractable, very achievable goal to treat those individuals that have this immune deficiency. And then in the long term, Jennifer and I have a goal to move beyond just monogenetic disease. We like to move into more complex diseases, things like cancer, solid tumors, difficult problems that haven't been addressed by current immunotherapies and current CAR T cell type therapies. And we think that CRISPR can play a role in that. But we also want to move on to neuro and neurological diseases. And I think there's a tremendous amount of opportunity when you think about the microbiome, when you think about direct editing of the brain, and you think about things ranging anywhere from mental health all the way to neurodegenerative diseases like Alzheimer's and Parkinson's. I think that genome editing has a tremendous opportunity space there, especially when you expand it to things like epigenetic editing, microbiome editing. I think there's real opportunity to make impact in these more complex diseases. Now, we do have an open sickle cell clinical trial right now. It's with UCSF, and this is the only nonprofit funded CRISPR sickle cell trial in the world. All others are startup companies looking to probably charge anywhere between a few $100,000 US dollars up to millions of dollars. Our trial is funded by the state of California. It's been funded by philanthropy and by federal government funds. There have been no private funds, and it's the only one in the world that's like that. But we're not going to stop with this trial. This is still most likely going to be an expensive trial. You have to take the cells out of the body. You have to do ablative therapies inside the body. This trial will still do all of those things. But as I'll show next, we think that there are ways to be able to improve this process. And Ross Wilson here, for those of you that here in the room, please talk to Ross about some of his efforts in editing directly HSCs, and I think pathways to be able to improve the efficiency as well as lower the cost of those treatments. Why is that important? It's important to go and try to ultimately achieve an in vivo delivery, a systemic delivery, where you're not taking cells out of the body, you're just putting the CRISPR reagents into the body. And those reagents would be smart. They'd be reagents that would be able to find the right cells in the right part of the body, and to be able to avoid many of the painstaking process that is involved in the current treatments. And I will say first that the current treatments are most likely going to get approved. I think it's in December of this year. Most likely, the first CRISPR-based treatment will be approved by the US FDA. So that will be a milestone landmark in the field of genome editing. But those treatments are not simple. They're not inexpensive. They involve taking out cells. They involve ablative therapies that oftentimes might sterilize the patient. Talking about weeks of hospitalization and lots of laboratory experiments to be able to grow up those cells to then transplant back into the patient. It seems to work. It's revolutionary, but it is by no means the optimal and ideal solution. So what you see in our current trial is a model after that. You do take the cells out. You do use electroporation to be able to get the CRISPR reagents into the cells. And then you grow those cells up, select the edited cells, grow them up, and then transplant them back into the patient. And most likely the cost will be something on the order of about $1 million. Okay. Ross Wilson, as well as our IDOR fellow, Bruno, have been working on a way to avoid electroporation, which is actually pretty bad for the cells. The cells actually, many of the cells die with electroporation. You can't actually do repeat treatments because of that toxicity to the cells. So we're now innovating ways to be able to deliver the reagents without using electroporation at all. You essentially just mix the reagents with the cells, and because of the specialized molecules on the CRISPR modality, it's actually able to just get those editors into the cells without having to use electroporation. And so I think this is a good stepping stone. And it also, I think, might be more applicable to countries that do not have maybe the cell manufacturing capabilities that the United States has. And I think that it's important to be able to take this step, both for regulatory and a scientific step. Just to show you an example of the power of these systems, this just shows two sets of data. One, I don't want to show very much data today, but I did just want to show that we can achieve very high rates of editing efficiency, both with like a Cas12, but then more importantly, a base editor that's actually editing the sickle specific mutation. So this is essentially an edit of the fetal hemoglobin that shows that you were achieving above the clinical requirement for efficacy by doing it with these molecules that Ross Wilson is developing. So how do we ultimately get in vivo? Well, those same molecules that Ross is developing, you can add on cell targeting antibodies to be able to help target the specific cells inside the body. So our hope is that as we keep refining these molecular tools, that you would truly just be able to take an intravenous infusion, and that those molecules would then not only find the right cells, but then also have a high percentage of efficiency of being able to move the reagents into the HSCs in the bone marrow. And it would essentially be a one-step process of being able to treat sickle cell, which is ultimately where we would like to go with this technology. We're not there yet, and many others are trying to get there, but it is a longer term goal of the IGI. And the hope is that the cost then would greatly and dramatically be reduced. And so across the board, Jennifer and I have a goal for the Institute to try to push in vivo delivery, because we believe across the board, it's a way to be able to help lower the treatment costs, the overall treatment costs of CRISPR-based treatments and therapeutics. Now, I did want to just show a little bit of data from Jennifer's group specifically. So Jennifer was not able to be here today, but this is I'm going to talk to you a little bit about a delivery technology that her group has been developing. They're called envelope delivery vehicles, and they borrow some of the molecular machinery from the lentivirus. The lentivirus has long been used as a vector to be able to help deliver genomic and genetic payloads. But what Jennifer's group has done, and to help with Jenny Hamilton, a postdoc from her group, is to take certain components off of the lentivirus that allow you to be able to go inside the membrane, to be able to sort of transition inside the membrane, and then also using these short chain parts of the antibody to be able to help target the specific cells that you want to target as well. So I'm going to show you data where they've actually been able to do an in vivo mouse experiment. So they used an intravenous injection in the mouse and use these envelope delivery vehicles to target T cells inside the mouse. Now, on the left-hand side, what you're seeing is there you go. You're seeing, now granted, this is 1.5% modification, but this is in an in vivo experiment where you're taking and you're targeting T cells using these envelope delivery vehicles, and you're essentially creating edited CAR T cells inside the mouse. And the efficiency, although it does not seem very high, 1.5% is actually pretty good for in vivo distribution. You probably don't need, Ross, you probably need what, 5%? It's a little bit controversial, but I think you probably need something on the order of 5% to really have therapeutic efficacy for in vivo editing. And with these EDVs, they're essentially showing that you can find the T cell inside the body, and that you can actually edit almost 2% of those throughout the entire system of the mouse. So it's a very, very exciting result. But what's interesting is that if you look at non-T cells, you actually see very, very low levels of editing. So the specificity of this is quite high. So again, imagine you're putting in reagents into the mouse. Those reagents could potentially edit any cell, but because of the molecular specificity that Jennifer and her group have put in, they seem to be editing T cells at a much, much higher frequency than those that are not. So this is just an example of where we are heading as an institute to be able to provide this in vivo editing process. All right, now I want to talk a little bit more about affordability. I've hopefully convinced you that technology in this area is a way and a path to be able to reduce, to be able to create better treatments, safer treatments, but also to be able to make them a more affordable option. We recently convened technologists, regulators, payers, insurance companies, and we've convened all of them, manufacturers, together to start thinking and planning and strategizing about ways to create more affordable treatments like those that I've talked to you about today. And we recently just published our report from our affordability task force. And so I encourage you, and we can provide the links and the informations back to IDOR so that you can start to understand some of the roadmaps, the possible options that would be there to be able to enhance the affordability of these treatments and therapies. And this is a task force that was led by our public impact team because it's such a priority for Jennifer and I for the Institute. Now, we also don't want to stop just at sickle cell. You know, there's 100,000 patients, potential patients in the United States, another 100,000 patients for sickle cell here in Brazil. But we also don't want to stop there. So we're starting what we're calling the IGI Center for Interventional Genomics. We want to be able to intervene. We want to be able to interface with patients around the world and essentially determine the editability of those diseases. Take their tissues, take blood samples, and try to understand what's causing the disease. How do those? How would we, as editors, as genome editors, potentially intervene to try to treat that disease? And to be able to do this in a way that it's not just one patient or two patients, but to be able to access a growing number of potential patients around the world. So we're going to have to look at things like how accessible is the tissue type, how we might be able to intervene to be able to more rapidly create these potentially life-saving treatments. And so this is something that's ongoing. We have a clinical molecular pathologist that's leading this effort, and he's going to be able to link patients or prospective patients directly to genome editors that then would try to work with clinicians to be able to truly change and save more lives. I've got a couple examples of this. We're looking, at least initially, at primary immune deficiency diseases. And we're looking at, you have many mutations, on the order of thousands of mutations that are out there that could be causing these rare diseases. And so we're looking overall at the ability to look at the VUS, or the variants of uncertain significance, trying to link and become more causative. So when you walk into a clinic, you get your genome sequenced, you have variations of unknown significance, and try to link them more specifically to the disease that you have, so that we then can move forward those candidates that are most suitable for genome editing tools and technologies. Most likely, many of those will be in the blood initially, because it's the easiest to access, like sickle cell. But I think ultimately there might be other tissues that we would be able to do this with as well. And so primary immune deficiencies are one of our first targets. And I mentioned this before. This is a collaboration between the IGI and the UCSF Gladstone Institute, where Brian Shai and Fyodor Urnov and others are involved with an effort that Alex Marson is leading on IL-2RA. This is a mutation that causes one of these immune deficiency diseases, and Alex Marson has an open clinical trial right now to try to treat IL-2RA. We also have an open effort with the team here between UCSF, Jennifer Puck, and Dr. Matt Kahn in the UCLA and the IGI to be able to treat this Artemis skid disease as well. And that really, I think, is directly in line with sort of the mission statement of what the IGI is doing, because this is not a disease that a startup company will would have the motivation to pursue, because it's such a rare and IGI. And so I will take the liberty of my presentation here and at least mention the other passion that I have for genome editing, which is to try to affect change on climate. And this is something I understand. It's not directly linked to health, but I passionately believe that climate change is also affecting global health, human health as well. But you may or may not know, there's a recent publication by the FAO, big, large international agriculture organization, that sums up all of the emissions that are responsible from agricultural, not just the growing of food, the growing of animals, but also the processing of food, the transportation of that food, essentially the entire impact in terms of greenhouse gas emissions from the fruit production systems. And it's almost 1/3 of all anthropogenic greenhouse gas emissions. 1/3. So that's the driving motivation for the IGI's climate and agriculture program. By 2050, we will have close to 10 billion people on the planet. At the same time, climate change is causing many, many more disasters. And oftentimes, those most affected by those disasters are in the poorest parts of the world. It's those that are being affected by storms, floods, droughts, high temperatures. We believe and have already started work on things like drought-tolerant rice, heat-tolerant, storm-tolerant, disease-tolerant. I mean, many of the South and Latin America are being threatened by many emerging diseases, for like fusarium wilt in the banana. We believe that CRISPR has the ability to engineer crops that are resilient to climate change, use less pesticides, use less water, become more climate resilient to help feed a growing population, number one. But number two, we also believe that we can actually create a net zero farm, a farm that has no net emissions. And that's would be remarkable. And contrary to the belief that most people have, those emissions aren't coming from the tractors and burning fossil fuels and all of these types of things. Most of the emissions are actually coming from biology. It's microorganisms in the gut of a cow. It's microorganisms living in a rice paddy that emit things like methane and nitrous oxide. Well, by doing that, we believe that we actually could intervene using CRISPR in those organisms to shut off or modify them so that they do not produce the greenhouse gases that are there. So we think there's a tremendous opportunity to do that. And I'll leave this presentation with just one note. Think about all of the green energy efforts that are going around the world. Everybody knows that, countries are putting more solar panels. They're putting more wind farms. At some point in the future, we're electrifying our transportation systems. At some point, fossil fuels and the energy industry are going to start to go down with respect to emissions. If we do nothing with agriculture, those emissions are going to continue to rise as our population increases by 20 or 30% more than what we have now. Do you want your food to be thought of as big oil? Do you want your farmers to be thought of as big oil and the petroleum industry? That is the direction that agriculture in the world globally is heading. Let's stop that. Let's intervene. Let's provide healthy, nutritious food with potentially lower emissions and in a way that's more sustainable. So with that, I'll leave you with this slide. This is our portfolio. We have efforts in all of these areas right now where we're using CRISPR and genome editing to be able to reduce farmer inputs like fertilizer and irrigation, improve crop resilience to drought and flood and disease, increase crop yield and nutrition, editing crops to make them more photosynthetic efficient, and to be able to increase the amount of biomass, but also reducing animal emissions, reducing crop emissions, capturing carbon in biomass and roots and in the soil. And if you can capture carbon in that soil, then you can also restore the soil health and take marginal lands and underutilized lands and perhaps stop deforestation, which I know is also a major consideration here in Brazil. So with that, I want to just announce that IGI had a pretty audacious idea about a year ago. We applied to the TED organization, the TED Talk organization. They run an audacious project every year. And we submitted an idea that said, look, we think we can actually transform human health and climate by directly editing the microbiome, not taking organisms out, not probiotics, not, just sort of slurping, shakes of probiotics, but by actually taking CRISPR-based editing modalities and using those as oral administration to alter the human microbiome, to alter the microbiome of a cow that's responsible for 10, 15% of all, agricultural emissions with methane emissions. And We received $70 million through this tech from 14 different philanthropists that believe in the IGI and believe in our ability to be able to do this. It is a little bit crazy. I won't lie. The microbiome is tremendously complex, and I don't know how much we will be able to control, but we are going to intervene early. We're going to treat calves. We're going to treat young children between the age of 0 and 2. And we believe that CRISPR has the ability to tune and tweak the microbiome to be able to offer lasting health and reduced agricultural emissions as well. So with that, I thank you all for your attention, and hopefully I've inspired you to pursue your own scientific and technological breakthroughs, because I really do think that science is the way to make big impacts in the world. So thank you all for your attention.
So for those of you who attended the research seminars a couple of weeks ago, maybe 3-4 weeks ago, I already gave an overview of what Pioneer Science is all about. But I think if you have seen our manifesto, our video manifest, as you saw it this morning, I think the connection between the IGI and the Pioneer Science motivations and mission and vision is tremendous. Really, I think both institutions, IDOR, Pioneer Science, and the IGI are really converging very strongly on the mission of improving human welfare through frontier science. So it is, I think, much more than the specific projects we're pursuing. This overall vision is what really unites the two institutions. So thank you, Brett. So as most of you know, no, let me go back to the first slide here. As most of you know, IDOR was born about 13 years ago, and it was created by the Moll family with strong support from the hospital company. And then about 3 years ago, its younger sister was conceived and born, the Pioneer Science Initiative. And this is a philanthropic initiative, as well as the IGI, nonprofit, with initial donations and gifts from the family. But we hope that by generating high impact science in the next few years, we'll be able to attract funding from additional donors and additional supporters as well. So that's the overall vision. And the tagline for Pioneer Science, if we need one, is really expanding the frontiers of knowledge, thereby improving the welfare of humankind. I just want to call your attention to a couple of lines from the manifesto that we wrote that describes our view of how we hope to contribute to this cause. And so the first one is that really we know that science moves in a continuous manner. Every day there are increments in knowledge about different fields, but occasionally, and not every day, there are these big leaps forward, these big jumps, like when Jennifer, CRISPR was already around, but when she figured out a way to really use this technique, this method to edit genomes, and that with Emmanuel Charpentier and David and Jennifer, that really led to a big jump in the biomedical sciences. So these are the kinds of leaps that we think unveil novel and wholly unexpected areas for discovery, of which we heard so many today from Brad. So that's really the motivation for Pioneer Science. We want to stimulate and foster novel approaches to scientific areas and problems that rise from unexpected or perhaps unorthodox questions or unexpected responses from Mother Nature that perhaps you would just sweep aside and say, oh, I don't want to look at this. This is a weird result. But perhaps in that weird result lies a really bold leap forward. So that's one of the inspirations for Pioneer Science. But of course, we don't want to put all our cards into only extremely high-risk projects. We want to fund those, yes, but we want to take a more rational approach of splitting a bit the investment into high-risk, high-return, and very frontier science with more mainstream, let's say, of science that is at the very edge of current knowledge in different fields. So it's headquartered here in Brazil, of course, And it has different ways that we hope to be able to achieve our goals. The first one is establishing partnerships with leading national and international institutions like the IGI, but I'll show a couple more examples in the next few minutes, both in Brazil and abroad. But we want to really look and target and collaborate, partner with institutions that share this vision, that science is not just for science. It has to have to be thought of as something that will eventually lead to improving the lives of our fellow humans. And providing, of course, since it's based in Brazil, we wish to provide outstanding Brazilian researchers with the opportunity for advanced training abroad. Yes, that's an essential and important component sometimes, but more than that, we want to make sure that we bring these talented young people back to Brazil so that we don't promote brain drain, but we reverse brain drain in a way. So we wish to provide the opportunity for exceptional training of talented, gifted fellows at leading institutions, but we can't force them to come back, of course, but we would like to offer them a sufficiently attractive environment or ecosystem, whatever you want to call it, so that they will feel compelled to come back and make a difference in our country. So that's the motivation for Pioneer Science. We currently have 13 fellows. This number is actually a bit outdated. We have a couple more here to add here already, fortunately, 2 of which are at the IGI. We heard from Bruno and Tiago today. They're here with us. And I should say that personally, I couldn't be happier about the progress that you guys have reported during these 7 to 8 months that you've been there. It's truly amazing what you've achieved. So congratulations again. We've said that before, but I wish to congratulate you again. We have 4 fellows that have benefited from a partnership with the Zona Institute, which is connected to Promega, the biotech company in Wisconsin. We have 3 fellows currently at the Weizmann Institute of Science in Israel, one that is on his way back from a 1-year period at Stanford, and then 3 more here at local universities in Minas Gerais and in Rio de Janeiro. These fellows have been training, and a few more, like as I said, there are a few more than 13 now. They have been training at 8 partner institutions, which I've listed down here. And we are also in an effort to promote the conditions and make the conditions better for the fellows to wish to return to Brazil. We're investing substantial amounts into building or rebuilding or renovation of labs here in Rio de Janeiro, here at IDOR, at the IDOR in Sao Paulo, the new IDOR building in Sao Paulo. There's a whole floor of labs that was recently set up. We've also set up one specific lab here in Rio across the street for the quantum vision project, which I will mention a little in a few minutes. And then we hope to be able to install CRISPR labs, both here in Rio, in Sao Paulo, and maybe Salvador as well. Yeah, for the continuation of the projects initiated at the IGI. So there are 3 large, main, broad target areas: molecular and cellular biology, neuroscience and cognition, and then the physics and mathematics of biology. This is an area that's clearly something that's emerging more and more every day. You know, the connections between quantitative methods, the application of quantitative methods in big science, big data science in biology. So this is one of the areas that we want to foster as well. So I wish to begin just to cover a few examples of what we've done so far in these 2.5, 3 years or so of the life of the Pioneer Science. I would like to begin by highlighting the partnership with the IGI that Brad just told us about. So these are the 2 initial fellows that I mentioned, Bruno and Tiago. Some of the PIs, the Brazilian PIs involved in the projects, and of course they have their partners. Ross is here with us. Fyodor couldn't come, but is online with us, I think. So he's at the IGI today. And it's really based on this inspirational drive of Jennifer Doudna to make the CRISPR methodology accessible to wider populations. So that's what we hope to achieve with the projects that these initial fellows are developing, but also with the upcoming fellows that will come in the future. This is just a reminder for for the IGI team here of our visit to the IGI last year. This is when we actually kicked off the partnership. And a few months after that, Tiago and Bruno were able to initiate their work there. So Jennifer was accepting here a nice inspirational sculpture showing this DNA double helix with the symbol of Berkeley and a jaguar as a symbol of Brazil, which I think shows this partnership in a very poetic manner, I think. So the two fellows, we've heard from them, as I said today. They show their reports for what they've done in the past few months and what they hope to achieve in the next few months until we hear from them again next year. And then I would like to start talking a little bit more about the other partnerships that we have now. So this is the USONA partnership that we have. This effort, and there have been a few students who have gone to the USONA Institute in Wisconsin, have worked there for a few months, and have been developing work in brain organoids and psychedelics. So this is one of the areas that is under investigation under this project. As I mentioned, we have this partnership with the Weizmann Institute, and these are the three initial fellows under the program. Eliana Ferreira is working in malaria. Felipe Ptak is working on biolubricants, and Paulo Cesar is working on Parkinson's disease. So they are all training at the Weizmann Institute, completing their training in some cases, and like Paulo had already been at the Weizmann for about a year and a half or so when we recruited him for the program. Eliana just went, so we're at different stages of training, but the hope is that they will come back to Brazil and continue their work in a close collaboration with the Weizmann Institute PIs, of course, but developing the science they are doing here in Brazil. Victor Guedes is a postdoc fellow who has been in Stanford. He was in the lab of Jeffrey Glenn at Stanford. Jeff is part of this CAMH Institute at Stanford, which is really very inspiring because it's an institute at Stanford that was created to really bring together medicinal chemists, synthetic chemists, and biologists, and medical doctors as well as research clinicians. So the idea is to create this environment where people with very different backgrounds, who perhaps might never have the chance to really talk, like what are the odds that a medicinal chemist and a clinician researcher will get together over coffee in the hallway and talk? Very little. So they have created this nice infrastructure called the CAMH. Jeffrey Glenn is one of the members of this institute, and Victor Guedes has been for a year working on novel antiviral agents, initially with the goal of developing antiviral agents for COVID. But it turns out that some of the drugs that they've been working on have more widespread antiviral activity, specifically against diseases which are of great importance to us, like Zika, chikungunya, and dengue, for example, which are these arboviruses which are endemic in Brazil. Another project is on one of the other large area that I mentioned is neuroscience and cognition, and we have now 4 fellows supported by Pioneer Science, with additional support from a grant from the Templeton Foundation, and a couple of a partner institution is King's College London, and of course the Templeton Foundation has been instrumental in this. A more exploratory or frontier area that we are exploring is the area of quantum biology, and that's an area that's emerging in science at the interface between quantum physics and biology, and the goal is to try to understand what biological processes cannot be properly or sufficiently explained with classical mechanical descriptions in physics. So you need to go quantum to really understand them. So that ranges from understanding photosynthesis to mitochondrial electron chain and ATP generation, to how birds fly and orient themselves when they migrate in the winter, to how turtles swim in the ocean and find their way in the ocean. So there are a host of different biological applications and field, not applications, but fields, questions that can be addressed by this. So this began as an institutional agreement with UCLA, specifically through Clarice Aiello, who's a young Brazilian assistant professor there at the department. She's actually an engineer of training, but she's been working very actively in this area of quantum biology. She runs her own quantum biology tech lab. And IDOR, through Pioneer Science, was one of the co-founders of this quantum biology tech lab in the California Nanosciences Institute. So we were trying to stimulate the development of this area, initially through UCLA, but more recently, we've actually moved to Brazil-centered initiatives. So one of them is the development of community in quantum biology. So we had a symposium last year here in Rio. Here's Clarice, Marcelo Souza, who's also involved with this. Paul Davis, who's a very prominent figure in biophysics and in this area, talking to us about the quantum aspects of life, whether life can really be understood without applying quantum concepts to it. And this was an event which was very really transversal in terms of the topics covered, as I said, ranging from photochemistry to birds to photosynthesis. So different areas of science were covered there. And we are now working with Marcelo and with Clarice. Especially Marcelo has been very active in leading this effort in Brazil to develop a community of young researchers interested in this area. So we're doing this through, for example, a web series that we just completed, 5 episodes. That's on the web if you're interested, describing in more or less lay terms and in a very relaxed, informal manner, what are the major questions, what are the major progresses that have been achieved in this emerging field. We're also working on developing an advanced school for students from physics, from biology, from chemistry who are interested in exploring this, understanding a bit more about this. So we'll probably hold this next year. And the effort, again, is to build a community in this area. One of the sub-projects within the quantum biology field is this one, which we call Quantum Vision. And that's a project that aims to investigate the role of quantum mechanics in explaining how the eye and the brain detect and interpret light signals. So whether we perceive light as a quantum phenomenon and we understand that in the brain, or whether we think of it only, we see it and interpret it only as a wave, as has been more traditionally thought of. So and then there are physicists from the Federal University of Minas Gerais, the Federal University of Rio, a biophysicist who works on visual system. Gustavo Rojencol is a younger investigator in São Paulo who studies psychophysics. So he specializes in psychophysical measures and tries to the goal of this really diverse team here, of which Georgia is a direct part, is to really understand the quantum properties of light in its interaction with the eyes and the brain. And I would like to thank you for your attention. This is really, I think, a very special moment for us to receive you here. We just had Alon Chen from the Weizmann Institute here recently as well. So we are very happy to host our partners here. And we hope that through this enhanced collaboration, we will come up with even more intriguing and exciting and inspiring questions for future research. So if there are now, I think we still have maybe 5 to 10 minutes or so, if I don't know, before the cocktail. So if there are any questions that anybody would like to address to Brad or to myself about the Pioneer Science, of course, we'll be happy to take them. Thank you.
Thank you so much for all this inspiring presentation. And this is probably a very basic question. Is gene editing only for very specific targets in very specific cells, or can we add something broader? Like, for instance, in sporadic Alzheimer's disease, we know that ApoE4 is a risk factor. Could we add it like broadly in infancy?
I think generally the answer is most likely yes. Can you now? No, you can't. You can't do it now. But we have definitely thought about ApoE4 as a potential target. But again, you have to remember, specific cell types, delivery to the brain is very, very challenging. So I think something like that is possible, but it is very challenging. But it is very challenging, and it's very difficult, and there have to be many steps to be able to do that. I think the other question you have to think about is, when would you intervene? And that sort of goes back to the question that we were talking about with public impact and ethics. And okay, if someone has ApoE4, very high risk for early onset dementia, when is the right time to intervene? If you intervene too late, it might be too late. Maybe there are already changes in the brain or changes in the microbiome that have already sort of set the course for that individual. So at what point would you have to intervene? And does society allow an intervention prior to even the detection or onset and symptomatic of a disease? And I think those are all questions that need to be answered to be able to safely think about doing something like that. So there are a few groups in the world that are working on this. And I think ultimately, maybe some point in the future, I think you'll see that be an option. But I think you do have to answer the questions about the ethics and the timing and how you would ultimately make it happen.
Thank you.
More a question about the vision. So we were talking about making new technologies more affordable to the general populations. And we are very far from that, because in the biomedical field, basically everything's on the big pharma hands. So how do you see this in the large scale? I mean, one is the science, the development of the ideas, but what about the line of production? What about everything that's involved in actually achieving large scale patients?
Yeah. It's going to take a little bit of everything. Okay. This is a technology problem. As I showed, if you could go to in vivo administration, I think that could potentially make things simpler, make things safer, and I think ultimately bring down costs. But I think there are also manufacturing challenges. Centralized manufacturing is expensive. You have to do very large batches, which makes the cost for reagents and the cost for cells very, very expensive and very high. I think there are ways to do distributed manufacturing, where you can do a localized system to manufacture. And there are a few companies in the United States that are trying to do sort of more automated, more distributed, more localized cell manufacturing platforms that include onboard sequencing, that include onboard QA, QC to try to do that. So I think manufacturing, so technology, manufacturing, but then it's also a regulatory problem. Is there a way to accelerate and speed the path to regulation so that all the data that you need for a pre-IND and an IND and then ultimately the clinic? Are there ways for certain cases where there's maybe compassionate use or other aspects that could potentially accelerate and then also lower down the cost burden to get to an FDA approval of some sort? And then finally, I think there's also a discussion that has to be done with insurance and payers. Is there a public benefit corporation model that you could potentially use that could be a public-private partnership to be able to help enhance the ability to be able to pay for these cures in a more affordable way? So the task force that I mentioned is exploring all of those. They're looking at many alternatives and improvements to manufacturing, alternatives and improvements to the technology and the molecular aspects of the technology and cellular aspects of the technology. But you also have to look at the payers, and you have to look at sort of the whole system. Obviously, as scientists, we don't have control over some of those things, but at least we can talk about and start the conversation on how to move forward, knowing that the objective is to lower those costs. But Sergio, you might have, do you have thoughts on this as well? No. Okay. Yeah.
So Brad, thanks for your presentation. It's always exciting to hear you talk. And I wonder if you have any thoughts on potential challenges that you may have in the regulatory aspects when you think about N of 1 studies and the human microbiome editing.
Yeah, I'll start with the second one. We also convened some regulators in the microbiome space as well. Not just FDA, but also USDA, the Agriculture Department in the United States, the EPA. For the microbiome, you have to understand that the end product of what's in your gut usually goes into the environment in one way or another. Just say it politely. So it's an EPA, Environmental Protection Agency problem as well. So when you are thinking about the regulatory aspects of this, you have to think safety first. I think that aspect of it might actually be a little bit easier for humans, at least, because you're editing microbes and there are billions of microbes, and those microbes are exchanging genetic information all of the time. So me going in and just making one tiny little precise change, I think regulatory agencies are going to be better with that. I think there might be a faster, easier pathway to that. But I think the containment might be a little bit more of a challenge. And so we're actually working, some of that TED Audacious project is to specifically work on containment strategies, oxygen exposure as sort of a kill switch, other ways that you can engineer a metabolic niche where the organism that you're altering is enhanced by the co-administration of a metabolite that then could give that organism a metabolic advantage. But then when it's removed from the system, pooped out, then that advantage no longer exists, and it would most likely be destroyed in the environment. So there's a lot of considerations in the microbiome, but we're trying to consider again a lot of those aspects. On the other side, a lot of people have written. Theodore has written volumes on N=1, rare diseases, neglected diseases, and whether there are ways to potentially sort of accelerate that process. And I'll go back to the compassionate use consideration. In some cases, there are no treatments. There are no options. And I think in those circumstances, the FDA will be in agreement that there should be easier ways to sort of get to a phase 1, N=1 trial where again, there is no option. Obviously, with the approvals of the parent or the approval of the patient, I think there might be ways to do that. Now, the more complicated question is genome editing tools as a platform. Can we get to the point where if it's the same Cas, modality, if it's a blood administration, and all you're changing is some slight, nucleic, I think that's a more difficult question. The FDA is going to have to think about that. There's a reason that all the safety testing and pre-IND, there's a reason all those things exist. Do no harm. You want to do no harm. So I think the questions are still going to have to persist as to whether you can speed up in those cases where there's a lot of similarity between the tissue type, between the modality that you're using. The FDA is going to have to answer those questions. And I think it's more challenging. I think there's an opportunity, but it will be more challenging because we want to do no harm.
Just catching up on this link here, I was thinking, of course, I understand there are regulatory aspects, but even for an N=1 disease, you need funding to develop the treatment. And it seems hard to me to imagine that you're going to be able to attract philanthropic funding for an N=1 disease. I mean, what's the association that's going to pay for that? So how do you expect to be able to raise the necessary funds for that?
Yeah. So it's a great question, Sergio. I think in certain circumstances, we will be able to raise philanthropic money to at least be able to initiate a clinical trial. And if it really is one individual, then perhaps we can develop a treatment to be able to do that, but that's not scalable. we can't do that 100 times, 1,000 times. It's not scalable. So, and I'm looking to some of my colleagues like Audrey, maybe, one of the strategies was to try to achieve, what's the name of it, Audrey? The accelerated review, or the FDA, what's it called? I can't remember. Accelerated approval. You can get these tokens that become very, very valuable. And if you're able to achieve one of these tokens, you then potentially could sell that to a company and use the money that you get from that to then fund additional trials and additional development of things. So that was one option. Another option was a public benefit corporation, where maybe you can raise private capital in some way. Maybe for countries that can pay for the treatments, they pay full price for the treatments, but then you use that money to subsidize the treatments for those that may not be able to afford it. So I'm not saying this as eloquently as I could, but there are many of these options that we're thinking about. We know the end goal that we should be going for, which is reducing these prices, making them more affordable, making them more accessible. And we want to try to work with Jennifer to be able to help make that a reality.
So just on the topic you're discussing, are there new models on, apart from VC-based models, venture capital-based, in which you have some kind of shared, that will require like an innovation in the business itself, right? Like a consortium of VC, could be government-based, but in which founders or scientists or scientist founders, the company will just subscribe to that model, like at a consortium. So you have, the upsides are much smaller, but you share risks, and eventually you could find solutions which are not typically VC backed. So therefore, there's no this big incentive for huge profits, and therefore the whole system might be driven a different way. I know this is very a dreamy thing, but is there anything along these lines being discussed?
I think there are examples of where you can get a big win. And by a big win, I mean an exit that produces $1 billion or some sort of large capital. The problem is that you can't predict when those are going to. So how many investments do you have to make before you get that big win that could then be reinvested to support all of the types of things that we're doing? So I think it does require a little bit of innovative business model to try to figure out how to maximize your chance of those wins that could then help support a whole ecosystem of innovation. And it's something that I think needs more discussion.
Yeah, especially looking at the index of biotech being negative now, it's very hard to, but when it's positive, who knows, right?
Yeah, we have Reed Jobs on our board, and I was doing a fireside chat with Reed just a couple of weeks ago, and he immediately said the environment is bad. I'm sorry, it's bad. It's for biotech, but he was still optimistic that the right technologies, the right approaches, there's still money there to be able to make investments, but hopefully it's going to bottom out and it'll start to come back up.
Hello, thank you for your presentation. I'm Rodrigo Viana from the University of Miami, and I run the transplant program there. And we use CRISPR technology for xeno transplantation. We're probably next July do the first clinical trial. You saw some organs going in, but disorganized. So I think we're going to do the first clinical trial. But my question to you is, we're looking into this in orphan diseases, right? Some diseases that affect 100,000 people here, and I believe that's difficult sometimes to raise the funds, but you have large scale diseases, people with diabetes, people with hypertension that produce nitric oxide in the wrong way, or people that have depression and because they don't produce tryptophan in the right way. Have you looked at broad spectrum, like, okay, let's help billions or millions, and maybe easier to raise funds? Have you looked into that? Is that is that possible?
Yeah, I mean, it's a great question. So this goes to the core of what the IGI is. I think, in fact, Ross, one of the very first conversations when I started working at the IGI with Ross was exactly this topic, N of 1 versus N of a billion. And I think there is no right answer, number one. But I think the best that we can do is to do both. Okay. A nonprofit, I think we would be neglecting our responsibility with Jennifer and with Ross and with Fyodor if we were not trying to help those that aren't going to be helped by big pharma, by startup companies. And if we don't do it as a nonprofit with the goal and mission of increasing equity and affordability, then we would be neglecting our role in this ecosystem. It's number one. But then number two, I'm an innovation guy. I ran the biotech office for DARPA. I'm never going to say no to big bets and big innovation. And what's bigger than things like obesity, diabetes, Alzheimer's? I would never admit that a technology as broad and as impactful as something like CRISPR or other genome editors, that there is not a role for this technology in those larger diseases. And I think our work in the microbiome is one example of that. The microbiome is implicated in all of the diseases that I just listed. If we actually could have exquisite and precise control and manipulation over the microbiome, I think there's an opportunity there. We can do direct injection in the brain. Ross just got some data from large animals that's showing that you can diffuse the CRISPR modalities throughout larger segments of the tissue in a large animal model through direct injection. We can start to think about going across the blood-brain barrier with some of these modalities and molecules. So the future is pointing in the direction that we can go after diseases that affect larger numbers of people. And then the last thing I'll say, there's a saying that goes on inside the IGI sometimes that human health is one application space that you might be able to affect thousands and maybe even hundreds of thousands of people, maybe even millions of people. But food could affect billions of people. So I have fought and will always fight at the IGI for the implications to human health, the implications to feeding society in a very responsible, very sustainable way, because I think ultimately you could touch the lives of every single person in the world if you could improve the way that we farm and provide protein and food to the world. So I think for us, I'm a grand, big thinking guy. I want to do it all. And we have to be judicious. And we're going to do some rare diseases. We've identified some early diseases that we can go after. We've identified some diseases that we might be able to work with in the microbiome. The TED Audacious, we're going to be working on asthma. Big target for disease. So we're trying to be judicious and not try all diseases. We're trying to be as focused as we can, and then also add in some of the agriculture and climate targets as well.
I think we have one last question in the back.
Thanks a lot for the presentation. It was amazing. I'm not sure if I misunderstood it, but I think you said something about epigenome editing. I was quite interested because we work directly with epigenetics here, my research group, and I just wanted to know, how can you exactly edit the epigenome using CRISPR, and if there are any ongoing projects at IGI regarding this?
Yeah, so one of our IDOR fellows is working on this specifically. So I encourage you to talk to Tiago after the meeting. He's been at the IGI and developing these technologies, but just in a really broad stroke, you can essentially use CRISPR modalities like a dead Cas or a dCas9 to sort of sit on the genome. And by doing that, it essentially controls regulation of that gene that it's in sort of partnership with. It essentially is a way to modify the transcription of the gene. So you're not cutting, you're just sort of regulating the gene expression. And there's a tremendous number of technologies that could do this. And I think there's a lot of potential. Some of those complex diseases that affect larger numbers of people, I think epigenetic editing is probably the future on how you can access and do that.
Right. Thanks a lot.
Well, I think it's about time we wrap up the session. It's been really exciting, Brett, to hear from about all the different directions in which the IGI is moving. Thank you so much for sharing your thoughts and vision with us. And I would like to invite all of us to move to the cocktail. There's going to be a small cocktail. Is it already up there? Yeah. Rosiane, our boss, is giving the thumbs up, so we can all move there and continue the conversation in a more informal setting. Thank you very much.
Yeah, thank you.